We now go on to talk about the final process in cellular respiration, oxidative phosphorylation. The oxidative phosphorylation process takes place in the inner mitochondrial membrane. Remember that mitochondria have two membranes, an inner and an outer membrane with a space in between. Oxidative phosphorylation has two components. The oxidative part, where NADH and FADH are reoxidized, and the phosphorylation part, where ADP is converted to ATP. Within the inner mitochondrial membrane, there are a series of protein complexes which act as electron carriers, and this is called the electron transport chain. As you can see in this diagram, there are three main complexes, named slightly peculiarly complexes 1, 3 and 4, plus several smaller ones, most of which we really don't need to consider, so are not shown on the diagram. The NADH and FADH donate their electrons to these proteins, lose the hydrogens as hydrogen ions into the mitochondrial matrix, and the oxidized forms of the coenzymes NAD and FAD are regenerated. The NADH donates its electrons to the first major complex, complex 1, while FADH donates its electrons to one of the smaller complexes, complex 2. The electrons from either complex 1 or 2 then move along the chain to complex 3 and then to the final complex, complex 4. Finally, this is where the oxygen is required. The electrons are combined with two hydrogen ions and an oxygen atom to make water. This process clears the electrons from the chain, and this is the oxidative part of the process. You may remember that I said earlier that if there was no oxygen available, the only part of cellular respiration that can continue is glycolysis, and now we can see why. The Krebs cycle converts NAD to its reduced form NADH, and the electron transport chain oxidizes the NADH back to NAD. This can only happen if there's oxygen available to remove the electrons at the end of the chain. Imagine a hose pipe with a blockage. If the water can't get out, no more will flow into the hose, and the same is true for electrons entering the electron transport chain. So if there's no oxygen, all the available NAD is quickly converted to NADH. So the Krebs cycle effectively stops because it runs out of NAD. If you are also studying chemistry, you'll be aware that electrons can carry different amounts of energy. This is important here because the electrons released from NADH and FADH have a lot of energy associated with them. As they move from one electron carrier to another, that energy is passed on to the electron carrier, so that by the time the electrons reach complex 4, they have left energy behind, if you like, in each of the major complexes 1, 3 and 4. This energy can now be used by the carrier proteins, and they use it to actively transport hydrogen ions from the mitochondrial matrix into the space between the inner and outer mitochondrial membranes. This is called the intermembrane space. So, as electrons pass down the electron transport chain, a large reservoir of hydrogen ions builds up in the intermembrane space. This creates a large hydrogen ion gradient across the inner mitochondrial membrane. As the membrane itself is impermeable to hydrogen ions, the ions can only get back across the membrane if there is a protein that can transport them back. Do you know how a hydroelectric power station works? You can see a picture of it here. In simple terms, water is allowed to flow from a reservoir to a turbine, and the power of the flowing water, which is driven by the mass of the water in the reservoir, turns the turbine blades, and that turning force is used to generate electricity. Well, biological systems used this process millions of years before the hydroelectric plant was invented. This is almost exactly the system that the mitochondria uses to synthesize ATP. This process is the phosphorylation part of oxidative phosphorylation. We now have a reservoir of hydrogen ions built up in the intermembrane space. This has created a large concentration gradient, and this gradient will drive the movement of the hydrogen ions back into the mitochondrial matrix, if they can find a way through. This is just like the water held in the reservoir in the hydroelectric power station. In the inner mitochondrial membrane, there is an enzyme called ATP synthase. It can allow the hydrogen ions back through the membrane. You can see a diagram of ATP synthase here. This is a big enzyme made up of a number of protein units with one segment embedded in the inner mitochondrial membrane called F0. You can see it in the diagram. A stalk, or axle as it's called in this diagram, and another segment which sticks out into the mitochondrial matrix, which is called F1. This is a really good example of a protein that has quaternary structure. The hydrogen ions move through the F0 membrane section of the protein, driven by the big concentration gradient across the membrane. And as they do this, they push this segment round, just as the water turns the turbine blades in the hydroelectric power station. The F0 segment is firmly attached to the stalk, so as it turns, the stalk turns inside the F1 segment. The stalk isn't symmetrical, so as it turns, it pushes against the inside of the F1 proteins, altering their shape. The F1 segment contains the active sites where ADP and inorganic phosphate, or PI, are converted to ATP. It's time to work out how many ATP molecules we can get for each glucose. From glycolysis, we have 2 ATP plus 2 NADH. From the Krebs cycle and the link reaction, we have 2 ATP plus 8 NADH plus 2 FADH. Now, the electrons from each NADH supply enough energy to make about 3 ATP. So 10 times 3 is 30 ATP formed from the NADH. The electrons from each FADH supply enough energy for ATP. So 2 times 2 is 4. So a total of roughly 38 ATP can be made by aerobic respiration of 1 molecule of glucose. Compare that with the 2 ATP that you get from anaerobic respiration. I told you it was inefficient. This explains why most living things, and certainly most cells in the human body, can only survive a very short time without oxygen. Cellular respiration is probably one of the most complicated concepts in A-level biology, so don't worry if you found this all a bit hard to grasp. However, it is one of the most vital processes in living things, so you do need to work at it. Here is a summary slide to help you get hold of the different parts of the process. You can see how the processes that we have looked at link together. The slide shows each process: glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation, and reminds you of the key function of each part of the pathway. If you've enjoyed this podcast and would like to do some more research for yourself, here's a question for you. Sugars, or carbohydrates, are one of the 2 main food groups from which we are able to make energy. What's the other one? How are these molecules metabolized to make energy? And where would this fit into our summary diagram? Here's a hint. All roads lead to acetyl-CoA. Once you are clear which process does what, then you'll be able to learn the detail that you need to know. So I suggest that you might find it helpful to make a summary table like the one shown in this slide here. It will help you when you're revising. Cellular respiration is vital for life. It takes a large number of individual steps to oxidize glucose and convert the energy within this molecule into the ATP which powers everything that a cell needs to do. But without that ATP, no living cell can survive.